Good evening, everyone. A very warm welcome to all our participants to today's webinar organized by the Center for Innovation and Precision Eye Health. My name is Wendy Wong. I'm a medical retina consultant from the National University Hospital, and it's my pleasure to be your host for today's session. Before I introduce our speaker, a few quick reminders to our attendees. Firstly, for awarding of CME and CPE points, please enter your full name. Secondly, we are interested in your thoughts and questions, so please share them with us through the Q&A feature that you'll find at the bottom of your screen. We will address these questions when we enter the interactive segment later on. It is now my honor to introduce our esteemed speaker, Dr. Nicholas Ponticos. Dr. Ponticos earned his degrees in computer science and bioinformatics before going on to pursue a PhD in machine learning at the University of Cambridge. He is currently Deputy Director of the Moorfields Reading Center and Clinical AI Lab. He also holds an appointment as Principal Investigator at the University College London Institute of Ophthalmology. He leads a lab that specializes in jointly analyzing genetics and medical imaging. Today, he'll be sharing with us more about his exciting, groundbreaking AI algorithm called Eye2Gene. This leverages ocular imaging in order to predict the causal gene for patients who have inherited retinal diseases. And this approach has the potential to revolutionize the way we approach diagnosis for this group of rare diseases. It is exciting to hear more about Eye2Gene from none other than Dr. Nicholas Ponticos, and I'd like to warmly welcome to invite him to begin his talk. Sorry, Dr. Ponticos, we will unmute you first before you proceed.
Thank you. Thank you very much. Thank you very much for the kind invitation. So I'm delighted to present our work to you today. So I'm going to talk to you about Eye2Gene, a AI algorithm to predict the causal genes from ocular images of inherited retinal diseases. So I'll give you first my financial disclosure. So this project is funded by the National Institute of Health Research, which is a funding organization in the UK, and it's funding this project for we started a year and a half ago, so we've still got a year and a half of funding left. We're halfway through the project. And also I'm a co-founder of a software company, which is unrelated to this work. So inherited retinal diseases, I imagine a few of the audience here are well accustomed to them, but for just a brief introduction for those who might not have heard of them before. So they are monogenic conditions caused by mutations in any of over 270 genes. And what you can see here on the left are retinal scans of patients which have a specific gene affected. So for example, ABCA4, EFMP1, USH2A, and choroideremia. Some people might have heard of ABCA4 retinopathies as more commonly known as Stargardt, or USH2As are a form of retinitis pigmentosa. But we are looking at individual gene level mutations, and that's how we describe them. In terms of conceptually thinking about how we see these different patterns of degeneration based on the genes, we can think of where the different genes are expressed in the different cells in the retina. And depending on where you're expressed, you get some quite characteristic patterns of degeneration, either starting centrally or in the periphery. I'll be interested in identifying the genetic diagnosis from a retinal scan, as I mentioned. So that means basically identifying the disease causal gene. So in the UK, inherited retinal diseases, although individually rare as a group, they are quite a common source of blindness in younger patients and working age adults, and they affect over 30,000 individuals in the UK. However, even at leading services for genetic diagnosis, we still have over 40% of our patients which do not have a genetic diagnosis. Inherited retinal diseases are a significant burden for the UK economy because these are lifelong conditions, and a lot of patients don't have a diagnosis, and those who do get a diagnosis, it can take a long time to get a diagnosis. So if you want more information about the impact of inherited retinal diseases in terms of socioeconomic impact in the UK, I refer to you to the below publication. So receiving a timely genetic diagnosis is extremely important. It benefits the patients in terms of their psychological support. It gives them an idea of a prognosis, and particularly helps them for their family planning, as it will tell them the chances of passing on this mutation to their offspring, for example. Also, there's quite some serious syndromic conditions that can be detected through a genetic diagnosis. So for example, conditions which might be affecting neurological conditions such as Batten's, for example, or some heart conditions or other types of syndromic conditions. And as we know, more and more genes are being targeted now with treatments. And this is a fast-growing clinical trial market for these patients. And of course, knowing what is wrong with the patient in terms of the genetic diagnosis can help the healthcare system to put patients on the right diagnostic path, on the right care pathway, so they get the right help. So just to illustrate the problem of the genetic diagnosis pathway in the UK, and this I'm sure is the same in many other countries. There is quite a lot of patients in the community and the general population. What is maybe more specific in the UK is that patients do come in, or I shouldn't say patients, I should say general members of the public come in routinely for eye tests at an optometrist. And this I know doesn't happen in every country, but in the UK, for example, optometrists do take retinal scans of people who attend the opticians, and so there's an opportunity there for them to detect something and then to refer to a specialist hospital. So these numbers are based on these numbers are based on our estimates in the UK. But as you can see, there is quite a large number of optometrists, but a much smaller number of specialist eye hospitals. So already we've got a bit of a bottleneck here. And then in each of these specialist hospitals, you have multidisciplinary teams. This is maybe, again, something which is maybe specific to the UK, but you have a team which combines clinicians, geneticists, bioinformaticians, and so on, working together to try and identify a genetic diagnosis for a patient. So even after all this process, we've got a genetic diagnostic rate, which is, this is based on the Moorfields number, which is around less than 60%. So you can imagine that actually, in reality, there's a lot more patients that are not being genetically diagnosed. So we're interested in trying to use AI to basically streamline this process and make it more efficient, to be able to detect and diagnose the inherited retinal diseases sooner. So we've written our work as a protocol that you can read here. There's a link here below to the protocol paper, which describes the whole project. So the funding we obtained, we are going to focus in this first phase of ITGene. We're focusing on within the hospital centers, how we can improve the genetic diagnostic, accelerate the genetic diagnosis. There is also a parallel problem, which is the referral problem, which is detecting it in the community. And this is something that we are starting to work on, but it's not something we have been focusing on so far. So my talk is going to focus on within the hospital setting, how do we more effectively genetically diagnose the patients using AI? So there's going to be two steps, which I'm going to two points where AI can be helpful, which is guiding genetic test prescription and guiding genetic variant interpretation. And I'm going to illustrate this with this flow chart here, which kind of shows you when a patient comes in with a suspected inherited retinal disease, the sort of pathway that they'll follow at the hospital. So important is to have a screening test, and that's, as I said, that's something that hasn't been the focus yet, but we're working on. Where ITGene comes in is to identify, assuming the patient is an inherited retinal disease patient, what type of gene might be causing that patient's condition. And this can be useful for prescribing the genetic test. And now we have different types of genetic tests. So we can look for more untargeted whole genome approaches or more targeted approaches, depending on what is the probability of a certain gene. And then when we get the results from the genetic test, we also have to identify and prioritize genetic results, basically the genetic variants that we receive. And this is also, we can also use information about the retinal phenotype here. So this is another point where ITGene can score genes based on their likelihood. As I mentioned, this is the first step, which is the predictive gene set. So you give ITGene a retinal scan and you predict what gene is likely to be causing the disease, and you get the probabilities here, and you can also get the recommended genetic test. And the second scenario I mentioned, which is you get the genetic results back, and then you have to put them through a genetic variant filtering pipeline. And this is another point where ITGene can help in prioritizing what genes to consider. So in order to build ITGene, we trained it on our data set at Moorfields Eye Hospital, which has over 2,000 inherited retinal disease patients. Actually, it has even more, but this is the number we obtained when we did the intersection of the patients with a genetic diagnosis and the retinal scans that we were looking for. So we were quite selective about the types of retinal scans. We focused on the Heidelberg imaging because that was the imaging device we had the most of. And then we focused on 3 imaging modalities, which is the 55-degree autofluorescence, the infrared 30-degree, and the OCT scan. And we also only kept the top 36 genes because we wanted at least 10 patients per gene. And this, I should say, was done quite a few years ago now. So we could, these numbers probably can be updated with some more training data. So this illustrates, as you can see here, the number of patients that we get per gene for each of the imaging modalities. And that's a total of 44,000 images across roughly 2,000 patients. The actual cohort of inherited retinal disease patients, not the imaging side, but just the number of patients with a genetic diagnosis, is described in this publication if you're interested. There is, I should mention, there's another paper in the works which is describing also the specific genetic variants for those patients. But this paper just talks about the gene diagnosis. So using that data set, we then split it into multiple folds in order to do something called cross-validation. This is described in the preprint paper that you can see here at the bottom of the slide if you're interested in learning more about the methodology. And so using that, we basically split the data set and we train individual networks, and then we use an ensembling approach to obtain an ensembled prediction. And now the reasons for doing this is because basically sometimes firstly we wanted to get some level of uncertainty. So doing this, you can see whether sometimes there's disagreement between the predictions, and that gives you some level of uncertainty. And another reason is because it allows you to do cross-validation. And the third reason is because also when you sample, you can also it's more robust to different sort of gene distributions that you can get in different populations. Because the gene distributions I showed you previously, those are in the UK population, but we know these can be different in different populations in the world. So after this, we basically have three. I2Gene is composed of three modules, three ensemble networks, which are specific to the different types of imaging modalities, one for fundus autofluorescence, one for infrared, and one for spectral domain OCT. And so if you have those three types of images available for a patient, you can put them into the I2G network, and you can obtain a combined prediction, as you can see here. Here are the parameters for the training of the AI model. As I said, it's described in the paper, so in the preprint paper. So if you want to have a look, please do read for further details there. So after we trained the I2Gene, we wanted to validate it. So we wanted to first we did some internal validation, which means we validated it on some held out data set from Moorfields. So this was working with Professor Michel Michaelides. So here's an overview of the data set that we held out for internal validation, so around 264 patients. And doing this, we obtained a top one accuracy. So out of 36 genes, how often does it get the first gene correctly of 66.7% and a top 5 of 85.6%. So I should say the top one doesn't seem particularly high perhaps, but you do bear in mind that this is having to pick out of 36 genes, and sometimes these genes can be quite phenotypically heterogeneous. And also this is significantly better than if it was just picking the majority class, which would be ABCA4. We also tried this approach we externally validated it. So we went to other data sets in different countries. So we took a data set from Germany, from two other data sets, from the UK, other sites, but in the same country, but two other countries, Bonn and Sao Paulo. And these are the results that we obtained. So overall, I2Gene was doing pretty well and was generalizing quite well. So this is always a problem when you're training AI systems, is that if you're overfitting on your data, you can end up thinking your method's doing really well, but when you apply it to other data sets, the performance drops significantly, and so it doesn't generalize well. So that's something we wanted to ascertain how well it generalized. And as I highlight here at the bottom, a completely random classifier would be 1 out of 36. That's maybe that would be a random classifier, but a naive classifier would maybe just pick the most common class, which would be ABCA4, which would be maybe a bit higher than 1 out of 36, but still would be significantly lower than 66%. It would be around maybe 30%. So we also wanted to compare this to human performance. So we know that this is a difficult problem anyway. But we did pick specialist ophthalmologists to do this. So with training in hereditary retinal diseases, and what we did is we presented them with images, unlabeled images, and we asked them to guess the correct gene. So unsurprisingly, this was a difficult problem, and so we didn't get a very high performance from this task. And also, if we combined the ophthalmologist results together, and we were quite generous, and we picked, if one of the ophthalmologists got it right, we picked that as a correct answer. We still didn't get an extremely high score. We got a 62%. And so this on the same data set, using only those images, I2Gene achieved 70%. So it shows that it's a difficult task for humans, and an AI is a good way of approaching this problem. Another interesting result is that if we looked at the specific gene performance, so what you have here is receiver operator curves, which are commonly used in machine learning and AI to show the performance of the system. So a perfect classification would be at the top left here. And what you are looking at is the area under this curve. So we are seeing this here for the different genes. The percentages are the percentage of the data set that these genes represent. What we are seeing in the different colored lines here are the performance of the different modules of ITGene with the different types of imaging modalities, so fundus, autofluorescence, infrared, and OCT. Firstly, I should say what we are seeing is that the genes like ABCA4, for which we have a lot of data, we're doing quite well in classifying them. And then genes like H2A and so on, we're still doing okay, but then as we get lower down, we're doing worse. The other interesting thing to point out here is that, as I said, the 3 different colors represent the 3 different imaging modalities. And what you can see here is how good are the different imaging modalities at identifying that gene. And for most of the top genes, you don't see much difference, but you start seeing some differences for genes like, for example, in particular CRB1, which causes thickening of the retina. So that's an interesting thing to see, that the OCT is doing better than the UNFAS imaging here. So this is illustrating what I just said. So thickening of the retina seen in CRB1 retinopathy, and OCT is doing a better job at helping us distinguish this gene. Also, what we, so this is using imaging only, but obviously there's other parameters which are useful, as we know, when we're trying to genetically diagnose inherited retinal diseases. And one in particular is age. So what I've plotted here is what you can see here are the genes, and on the bottom are the ages of our patients in our data set. So this is just plotting our data. So red means high density, so lots of patients in that region. So for example, for CACNA1F, we get younger patients, whereas for these genes we tend to get older patients. And I should say this is the age at first presentation. So this is not necessarily the age of onset, but the age of onset is not easy to get, so we're using that. So that can be useful, and it also shows the spread of the ages depending on the types of genes. And another one, of course, is the mode of inheritance when known. So mode of inheritance a lot of the time is unknown, because the patients might come quite late and we don't have their family history. But if they come in earlier and we do have their family history, then we can get a good idea of their mode of inheritance. And here we are seeing different, sometimes we have some genes, obviously, that can be multiple modes of inheritance, and that's difficult, but some of them are very specific, like the X-linked genes, for example, RS1, RPG, RP2. If we know it's an X-linked condition, then obviously that limits the search space for the possible types of genes. So that's very helpful. So if we combine, if we add the age of the age of presentation and the mode of inheritance into the classifier, into the final sort of classification prediction, then that is helping a lot ITGene in a few cases. So what you've got here, the different colored lines, age, a model that only uses age, only uses mode of inheritance, the ITGene model, and then red, the combined model. So the red line is always doing better than the green line, which is using only the imaging. And obviously, if you're only using age and mode of inheritance, you don't do very well by itself, which is what you would expect. So that's, if you have those parameters, obviously that's useful to include in the ITGene prediction model. Another interesting result from ITGene is that it kind of recapitulates what we know about the phenotypic similarity between genes. So what I've shown here is what you're seeing here. Each gray point is basically the result of putting an image into ITGene and then projecting it in a 2-dimensional space. So when an image goes into ITGene, it gets processed by different layers of the ITGene network, and this ends up at the penultimate layer just before it gets a prediction. You have a quite high dimensional vector. Basically, it's 1,024 dimensions, and that kind of gives you a very high dimensional representation of what ITGene understands this network, this image to represent. And then if you project that onto two-dimensional space, which is what we're seeing here, each point is projecting on two-dimensional space, you can deduce this to give you some idea of how similar to ITGene the different input images are, if that makes sense. So how close they are in the two-dimensional space is how similar ITGene thinks they are. So the red points are, each red point is an ABCA4 retinal scan that has been put in for here, and for H2A, these are what the red points are, and you can see, and so on. So you can see, for example, that most of the ABCA4 points cluster together, the H2O points cluster separately, and so on. And you can also see sometimes there's splits in the spread of the data, which kind of shows you that there might be two different phenotypes underlying this gene. So using this and clustering the points, we can obtain what you've got here on the right, which is known as a dendrogram, which is a way of showing the clustering and the similarity between the classes. And using this only on the 36 genes that ITGene has been trained on, we see some clustering of Stargardt genes and achromatopsia genes, for example. And what is really interesting is that we can extend this to genes that ITGene has not been trained on. So here we've got 69 genes, which is all the genes that we've got at, well, no, sorry, it's not all the genes we've got at Moorfields. We've got 123, I think, genes at Moorfields. This is like, but a bigger subset of the genes we have at Moorfields. And using that on ITGene, which has not been trained on all these genes, we can also see clustering of phenotypes that we know about. So here's a clustering. So the achromatopsia genes, the Stargardt genes we talked about, some LCA genes in yellow, Leber congenital amaurosis, some RP genes, and some ocular coloboma genes. So that's an interesting approach to kind of identify phenotypic similarity in a quite data-driven way, which doesn't rely so much on human interpretation. So there are still some challenges. I've kind of touched upon this, that the data set is quite imbalanced with a lot of ABCA4 patients represented in our data, and then a lot more rarer classes. And this reflects in the accuracy at predicting these classes. So more, if you have more training examples of you do better. And what tends to happen as well sometimes with these networks, these neural networks, is they tend to cheat a bit in a way by sort of over predicting the common classes and then predicting the rare ones. So this is something that we are addressing with more data and trying different approaches. So one approach we tried to address this was to generate synthetic data. Obviously, if you have more data, that's the best way of doing it. But if you don't have more data, then we thought synthetic data would be an interesting way of going about it. So what you've got here are each of these rows are synthetic images generated for these for these genes. And what we found and published is that although we could generate pretty realistic images, it wasn't really improving. When we were adding these images, we were not really improving the performance of ITGene. But a result we did find interesting is that if we only use the synthetic images, even though they are different, obviously, than from the real images, we could still use them to train ITGene to a pretty good level of accuracy. So that's an interesting outcome of this, even though it didn't improve the overall performance of ITGene, combining the real images and the synthetic images. And another approach we're taking is to look at more fine-grained features that we can identify using automatic segmentation algorithms in the images. For example, some of these features which overlap with other diseases, such as losses of the RPE or the ellipsoid zone loss and so on, trying to quantify that and then seeing whether we can find some gene correlations to help improve the classification accuracy of ITGene. So this is work being conducted with the Moorfields Reading Center, led by Konstantinos Balaskas. And we've got some apps that we are, which are being used actually, as I said, for other diseases, like for example, geographic atrophy, that we're repurposing for inherited retinal diseases. And we've deployed a demo version of ITGene, and there's an app online which you can, if you want access to it, just send me an email, and we're happy for people to start, trying it and give us some feedback. And we've also got a system at the Reading Center which allows uploading of retinal scans if people want to join our ITGene project, because we're always looking for more data sets of inherited retinal diseases across the world in order to improve the performance of the algorithm. So that's what I'm showing here on the right. This is our portal where you can upload retinal scans and so on. In conclusion, ITGene is able to predict the 36 most common inherited retinal disease genes in the populations that we have tested to the top 5 accuracy of over 75%. The performance is similar, and I would say better than a consensus of human experts. However, we still need to increase the dataset size, diversity by looking at other populations, and working on AI interpretability, which we're working on with the identification of features. One thing I haven't really touched upon is disease stage. We are touching that a bit with the age at presentation, but some diseases can be, progressive diseases can look very different depending on the stage of the disease, and tend to all look quite similar towards the end, obviously. So that's something to consider. And we're focusing on the Heidelberg device, but we're very much aware that this is a device used in specialist hospitals and is not commonly used elsewhere, especially in the community. So we need to start working on other devices as well. And we're hoping, by the end of this project, we will have ITGene in a state where it can start being used, at least under the supervision of specialists, and we'll be able to democratize and give wider availability to inherited retinal diseases, inherited retinal disease diagnosis services in the world. We have a website. You can have a look. We're updating it with our various publications and so on. So please have a look on our website. So yeah, thank you very much. I'd like to thank all my collaborators at Moorfields, UCL, obviously all the other external sites. And in particular, I would like to give big thanks to all the patients and families for allowing us to conduct this research. So thank you very much.
Thank you, Dr. Pontikos, for a wonderful talk where you've really walked us through some concepts which are very complex and technical. And as we begin our interactive segment, I'd like to invite members of the audience who have a question to please input it into the Q&A box, which you'll find below. And as we wait for questions to roll in, allow me to introduce our 2 panelists that we have joining us for today. First of all is Dr. Tham Ee Chong, who is an assistant professor at the National University of Singapore. He has a PhD in ocular epidemiology, and he's currently driving a lot of AI-related projects. Our second panelist for today is Dr. Chan Hui Wen. She is the lead for Inherited Retinal Disease Service at the National University Hospital. She has been trained in both pediatric and adult ophthalmic genetics at the Moorfields Eye Hospital, as well as the Ghent University Hospital. So may I now welcome comments from our panelists.
Thank you, Wendy, for the introduction, and thanks very much again to Nicholas for the wonderful talk. Absolutely exciting new frontier. So many congratulations on your exciting, innovative work. And if I may, I would like to start with one question. So what's exciting to see is the improved accuracy deploying i2Gene compared to the conventional model where you require a specialist, or rather a group of multidisciplinary specialists to diagnose the gene involved. So that's very impressive. And I would just like to ask, moving forward, if you are allowed to share, are there plans to further evaluate in terms of actual costs saved using i2Gene model versus conventional model? The perhaps an estimation in the total saving in terms of resource utilization, and the more tangible benefits to the patients as well. And I'd like to hear your thoughts on this, Nicholas. Thank you.
Great. Yeah, thank you very much for the your interest and for the great questions. I think saving costs is absolutely important, and to demonstrate that, especially for the healthcare system, if they are going to be funding the use of i2Gene and its integration in the healthcare system. So we have a work package dedicated to trying to evaluate this, so the health economic work package. And our approach is going to be to run some surveys, and I've been talking actually to Dr. Wendy about this as well. And we are running surveys. We're planning on running surveys in the UK to understand the standard of care of inherited retinal diseases across the different hospitals, both from the doctor's perspective, but also from the patient's perspective. So we understand both sides of the coin. And then I think so that will give us an idea of, what kind of, what is available, and how long. We already have some data of how long it takes to get a diagnosis, but it'll give us a better idea, and all that will give us an idea of where i2Gene is the most likely to be useful in the care pathway. And then in terms of simulation, we need to also run some simulations, I think, and actually use i2Gene, start using i2Gene at least in-house. We can't really use it, it's not a medical device yet, so we can't be using it obviously for patient care, but use it as a sort of silent parallel service system that we can use to evaluate, okay, and see, okay, i2Gene has predicted this. Now, what would happen if we follow the i2Gene suggestion? And we can do that with our prospective patients, obviously not letting it influence the standard of care for now, but seeing how it would basically hypothetically seeing what the impact of it would be. Would it, if we follow that advice and went for that gene, that gene panel, for example, or if we went for that gene line, would we have saved time rather than going through a whole genome process, for example? So that's something we need to we need to start doing in-house, I think. And I should say another thing I found on the variant interpretation side. So once you get the results from your genetic test back, people have been using these genetic variant prioritizers like Exomizer. I don't know if anyone's heard about this. Basically, you input these human phenotype ontology terms, which are descriptive text phenotypes. So these obviously are limited because they're limited by what is known about the associations of the HPO terms with the genes. And actually, I found from my work, that there's not a very good, there's a lot of missing gaps there, because they expect there's a lot of work that needs to be done to curate that data set, basically, to get those associations. And actually using an image would be a probably better way of doing it than using a descriptive phenotypic term. So sorry, that's going a bit beyond your question about health economic evaluation, but I thought that was an important point to make about the variant interpretation side of things as well. Sorry.
Okay, great. So we have a number of questions coming in from the audience, and we have one from our colleague who also sees IRD patients from SNEC. His question is about the imaging modality that's currently used for i2Gene. It looks like it's up to 50 degrees. And what happens for patients with RP where the disease manifestations are more peripheral? Perhaps Dr. Ponticos, if you could comment.
Yeah, so that's a very good question. I think we started with where we had lots of data basically, and that's what we had a lot of data on. We had a lot of data for this Heidelberg imaging, but I understand that wider field, and especially Optos and other imaging devices, I think Zeiss as well, are now more and more being used for these patients with peripheral changes like in retinitis pigmentosa. So we need to extend the model to be trained on these types of images as well. But it would require further that data set to be put together and then training on that data set. It wouldn't work kind of out of the box on that data set because these are not images that have been seen before. I mean, we could. I guess we could try it just on those images, but I don't think it would work particularly well. It would need some retraining. Yeah, it's a challenge, I think, and this is not only for ITGene, but it's even more pronounced than ITGene because we're working on rare patients. But it's a general challenge that this standardizing imaging in a way that we can use it for training is, something we need to really consider. And a lot of times, the imaging is not really for an AI; it's more for the doctor. And so it might not always be very standardized.
To add to this question, can I ask why is it that fundus photos were not included for the ITGene analysis? Because this is still one of the imaging modalities that is still more accessible.
The color fundus images. Yeah. So that will be in the stage where we are looking at the screening, I think. So I mean, that's a good point. Our project is quite, I mean, even though we're trying to include other sites, it's quite UK centric. And we just happen to have lots of Heidelberg imaging. Color fundus imaging, from my understanding, usually that's the first test. Everyone gets that done, right? And the opticians and so on. So that would be a really important to develop that, to use that for the screening model, I think, for ITGene. And then maybe we have we have basically two ITGenes. We have the ITGene, which is a screening step. And then we have the second step ITGene, which is the working on the more specialist imaging and on the Heidelberg imaging as a second step. Well, we'd assume that most specialist centers have more than color fundus imaging, although, as I said, that's maybe true for hospitals in Singapore and UK, but maybe not for maybe other countries where they don't have maybe these more expensive devices. So again, I think we're open to that. It comes down to having the data to train the algorithm on. So our next step would likely to be working on TopCon imaging, which provides a fundus image. So we would try that as well. I don't think we'll have very big numbers, but we will try that. Yeah.
Okay, great. The next question that we have is a bit more specific to genetic sequencing. So the question here is, what are our approaches for genetic sequencing and whether there's any difference in what we're doing here in Singapore versus in UK? So perhaps I could invite comment from Dr. Chan first.
Firstly, I would just like to thank Dr. Ponticos for this amazing talk. I'm sorry to have joined late and miss a great chunk of your talk, but I have some questions that I'd like to ask you shortly. So in brief, well, there is targeted sequencing, there is panel testing, there's whole exome, and then of course the now whole genome sequencing. As a first line, I think most centers would opt for panel, a curated IRD panel, or whole exome sequencing. And then based on the findings and based on, so there's also interlab variability, and then the diagnostic yield will also vary accordingly. But these two will be our first line. and then based on what you find, and then you can decide on subsequently just targeting one particular gene, looking particularly if you've got some missing heritability, you only find one variant, you might want to look in the intronic region, or then proceed with whole genome sequencing. I hope that answered the question. So Dr. Ponticos, some questions for you. I'm sorry if you've already mentioned this and I missed it. I was wondering how the team came about to select 36, those 36 genes. Is it because they have more macular-centric kind of initial presentation? And also, I think the diagnostic yield of 75% is really, really incredible. Is this for the top 1% hit, or does this refer to the top 5% and it's all accumulated together?
Yeah, no, great. Thanks for the good questions. Yeah, so 36, 36 genes. It was actually based on the data that we had available. So we put a cutoff at a time on having at least 10 patients per gene. So that's how we came up. But actually, we're increasing that number now because we've got more data and we've done another export of all our images. So we're increasing that number. So yeah, it wasn't based on the actual phenotypic appearance of the gene. And then, yeah, great question about the accuracy. So this is using the 75% you're quoting is based on the correct gene being in the top 5, not being in the first gene. The actual, at the moment, we're always working on trying to improve the performance, but at the moment, I think it was around 60, the number I gave you was around 66, 68% for the top 1 gene, which is doesn't come across as being very impressive, but it is a difficult problem with a lot of phenotypic variability and a very unbalanced data set. But we are still waiting. We're still working on making this better, because I think the work, going back to where you described the sort of process for genetic testing, right? Of like going through these steps. I think we do need to be careful, and this goes back to the question about the health economic aspect as well. Is we don't want this to be sending people down the wrong path, right? And then you end up testing this gene and thinking, oh, it's this gene, and then spend more money on the genetic testing in the end because we've gone for a different path. So that's something we need to be very mindful of, which is why we need to do this sort of analysis. So yeah, I think does that answer your questions?
Yep, it does. Thank you. And if I just may ask one more question. So given
Sure.
the phenotypic variability as well as the phenotypic overlap, I think what you showed just now with the PRPH2 was quite impressive with the 2 clustering, because we know that it is the great mimicker of all the IRD genes and phenotype PRPH2. So how does the I2 gene kind of is able to break through that, given that there's so much phenotypic overlap, to then be able to even get the top 5 hit for, these are going to be the top 5 hit for this particular phenotype?
Yeah, so it is a bit, at the moment as it stands, it's a bit of a black box system, because you just put in the genes with the label, with the gene, the images with the gene labels, and then you give it many of these examples, and you train the network, and then it gives you the classification output. So it's difficult to know exactly, what's going on under the hood. One way of doing it is, as I showed you, these sort of plots where you can kind of see what it thinks the similarity between things are. So it can give you an idea of the sort of clusters that exist in the data as far as it can tell. Another way, which I didn't really show, is called saliency maps, but we didn't, to be honest, we tried those saliency maps and they are in the preprint. I'm not sure if they're in the online version of the preprint or one of the other versions of it. But anyway, they should be in the preprint. If not, just ask me, I'll send it to you. We didn't find that they always work very convincingly the CNC maps, and it's quite difficult to interpret. Yeah, so that's it. So that it doesn't really answer your question, but it is a bit still of there's an element of mystery still, I must say, on how it's doing things internally. Once we start also looking at the actual features and segmenting features, which I mentioned, we're looking at fluid or looking at loss of various layers of the retina, like the ellipsoid zone and the RPE, that we can start also, we can use that as a, because these are quantifiable measurements, so we can say, okay, we see more of this type of pattern of retinal measurement in this type of gene, and then that can give us some interpretability. But we're going to lose a bit of the sort of non-interpretable aspect, which is also useful in doing things in a more objective way, if you see what I mean, like without you going for the features that we know about already. Ideally, what AI will like it to discover features for us, right? So I think that's certainly something we should try and work towards. And to do that is, yeah, I think CNC maps is one approach, but the other maybe approach like sort of radiomics approaches, where you might want to segment lots of random features in the images and then try and find some correlations from that. But that's also not very easy to do. So yeah, it's a bit of a mystery still, I guess, at the moment, how ITGene is doing it.
Okay, great. So I think we've covered some of the more technical aspects about ITGene, and I think we have time for one more very thoughtful and practical question from our audience on how early discovery of inherited retinal diseases would impact health promotion interventions within the country. So I'll invite Dr. Ponticos for comments first, and then if we have time, Dr. Chan and Dr. Tan.
Yeah. So if you can only treat what you can diagnose, right? So I think a lot of people are like, well, what's the point of diagnosing these patients? You can't treat them. But I think you have to start somewhere, and if you can diagnose them sooner, then you can obviously add to the knowledge, to your clinical knowledge base. It also motivates the economics of these clinical pharma companies that would be doing these clinical trials and so on. So they're always looking for patients, the pharma companies, as people who work in this area know. So if you can identify patients, more patients, then they will put more money into developing these treatments. And obviously, it will also drive the price of the treatments down for patients if you can identify more patients.
I think
and there's the therapy window as well, but I'll let maybe Elvis can talk about that.
Yes, I think those are
Sorry.
great. And maybe Hui Wen, if you could also add to that more about the therapeutic window.
Thanks, Wendy, and thanks for the great question. Certainly for the 300 different genes causing IRD and to a certain extent optic neuropathies that we know, they all have different varying age of onset and varying speed of disease progression. So the more we understand about a particular gene and a particular condition, the better we can curate and time the therapeutic intervention. So I think Dr. Ponticos has summarized that very nicely. The other thing we need to know better is the socioeconomic burden of an individual living with inherited retinal diseases. And I'll point you towards Prof. Robin Jamieson's paper that she put out recently, where they use a microsimulation model to try and study how much it costs for a person to live with IRD with severe sight impairment, and it's something around like 6 million Aussie dollars. So it's not insignificant. So if you think about treatment, if treatment is $1 million and you return functional vision to a patient, the cost saving could be significant. So that's all I have to say to that.
Thanks, Hui Wen. And so before we close the session, I'll also invite brief comments from Yi Zhong.
Sure, thank you. I think Dr. Ponticos and Dr. Chan have very nicely summarized on that point. There is definitely a very positive impact and benefit when it comes to earlier diagnosis in this context. I have no further comments to add. However, I do have one more question for Dr. Ponticos. So given that your I2G model was mainly trained based on your UK data set, do you have plans to further validate this in Asian population, given the genetic variations across different populations as well? Any thoughts on this, Dr. Ponticos? Thank you.
Yeah, absolutely. Thank you. Yeah, another great question, and absolutely, we're working with Prof. Kaoru Fujinami on this, and we're trying to get a data set from the Tokyo Medical Center there to basically validate it there. So yeah, absolutely. And if anyone else is interested in joining, I would be very happy to discuss. We are, just to update you on where we are with the project. So as I mentioned at the beginning, we are halfway through the project in terms of the funding, and we are presently working on starting to write grants to seek further funding for prospective validation in further data sets, so in further patient cohorts. So if you're interested, we could try and include you on this grant we're writing. So we could put you as an extra site. So please do get in touch if you're interested. Yeah, thank you.
Oh, most definitely. Thank you, and we shall touch base very soon.
Great.
So thank you, everyone. So as we conclude today's webinar, once again, a heartfelt thanks to Dr. Ponticos for taking time to speak to us at today's webinar, and also to all our attendees who've taken time out of their busy schedules to be a part of our webinar. We will now show you our slide for our next upcoming talk, which is in a month's time. You can find more details on our website or by using this QR code. Okay, so wishing everyone a good evening, and we look forward to seeing you at our next webinar.